So basically, the overlap of ecological niche is how yeast compete for the available limiting nutrients, limiting resources in the fermented beverages and the raw materials for the fermented beverages, such as that the wine must for the production of wine, the wort for the production of beer, and the cider must for the production of cider. One major challenge with the production of fermented beverages is that to avoid spoilage, we rely on the addition of chemical additives, such as that of sulfites. On the other hand, there is an emerging strategy which is called biocontrol, which is not yet fully understood and it's not predictable. So my project is trying to find ways to use biocontrol in a more mechanistic way, to find new strains that are predictable to be used for the prevention of spoilage. My name is Athanasios Kambardonis. I come from Greece, and I'm a PhD student in the DC7 project of spoil control. My academic background started with a bachelor's degree in alcoholic beverage technology and enology. I have mainly been focusing since then on applied food sciences. My project is about the study of the superposition of ecological niche of yeasts. And I'm basically studying how yeast compete and interact in fermented beverages. I'm in the phase of conducting pre-experimental trials for the main experiment of my project. During this time, I'm preparing synthetic media that mimic the real conditions of wort, wine must, and cider must. I'm preparing standardized yeast pre-cultures, and I'm also trying different combinations of yeast species and yeast strains to see what works and what doesn't. At the end of each experiment, I'm also analyzing the cell viability to see the effect of a potential biocontrol, which can be structured either by a singular yeast species or maybe with microbial community. I'm also sampling both substrates to study through metabolomics the effect of metabolic inhibition during the growth phases of the yeast species. One technical challenge that I'm facing is reproducing as faithfully as possible the substrates in controlled laboratory conditions. The utmost goal of this research is to develop biocontrol strategies that can be implemented by the industry and can be used for the bioprotection and lead to more sustainable fermentation practices. Other sectors can also benefit, such as that, in the broader sense, microbial ecology studies. Ecological overlap is strongly related to how yeast communities or how microbial communities assemble, how stable they are, and also whether introduced strains can establish themselves or not. These questions are also relevant in environmental microbiology and agriculture.